Hi everyone, this is Dr. Karen's, and in this section or this video, I'm going to be talking about the results section of your unknown project lab report. So in this section, you are presenting the results of all of the experiments that you did for this project, and that should include your gram stain. I want this information presented as a table, and this table should look familiar to you because this is the table I had you fill out during the practice run, and it's also the table I gave you to fill out for your unknown. But you're going to be recreating this table for the paper. So what's in this table? Let's remind you the name of the test, your observations, meaning what did your results look like? So what did the test look like when you were done with it? What color? Was there gas? Is there a clear zone? Whatever you're looking for um, in that test. And then what is the result? Is it a positive result or is it a negative result? Some of those tests have a slightly different notation that you're going to use for positive or negative, and so you need to pay attention to that. I'll talk about one specific test in a minute. And then what is your interpretation of your observations? That means what does positive or negative mean? So let me give you a couple of examples. So if you're reporting your gram stain results, the name of the test is your gram stain, right? Gram stain. Your observations will be, what do you see under the microscope? Do you see pink rods? Do you see pink coxy? Do you see purple rods? Do you see purple coxy? And then for your results, that will be either gram positive or gram negative, and you can include the morphology as well. Your interpretation of a gram stain would be if you see purple, you see that's gram positive, that's your result, and your interpretation would be thick peptidoglycan layer. If you see pink, your result is gram negative, your interpretation would be thin peptidoglycan layer. For one of the biochemical tests, let's use phenol red lactose. So your observations will be, if you remember, that test is the broth is either going to turn yellow and there may or may not be gas produced, or it's going to stay red if we're kind of staying simple. I guess a dark pink is an option as well. Those are your observations. That's what you're looking for. What did you see? The result, this is an example of a test that's not just plain positive or plain negative. Phenol red tests have different notations for their test results. So, a positive test result is one that turns yellow with or without gas formation. So, the more precise way to report that result is A or AG. A means acid, and the acid is produced during the fermentation process, and that turns the broth yellow. So, did you get acid only, yellow only, or did you get yellow and gas? If it's yellow and gas, then it's AG. Both of those are positive test results, but we have a little more specific way to denote that for specifically phenol red tests. Another test that has very specific notations for the results is TSI. You need to look at the TSI table in the lab book for that correct notation. Um, so results, we talked about results for phenol red. So interpretation. If your phenol red lactose tube was yellow and had gas, you have an AG positive test result. What does that mean? That means that your bacteria was able to ferment the lactose in the tube and it produced gas as a result. That's your interpretation. If the broth stayed red, that's a negative test result. Your microbe did not ferment the lactose in the tube. That's your interpretation. Okay, so you're going to be doing that for every test that you conducted. This table needs to be regenerate. It needs to be a brand new table. You cannot just scan the table. If you filled it out, the one that I gave you to fill out as you were doing your experiments, you can't just scan that and include that in here. You need to recraft your table. It has to be digital, so it's neat and I can read it. Also important, you need to report the results for every test that you conducted, even if the results ended up not being that relevant to your identification process. If you did the test, you need to report the results. This has to include your gram stain. This means that whatever tests are in your results table should be in your materials and methods table as well. Both of those tables should have the same exact tests. And like the materials and methods section, you are not just giving a table here. You have to give a few introductory sentences that tell the reader what they're about to see. What I don't want you to do not describe your results in paragraph form. All I want is the table filled out as instructed. Do not include any description of methods. Don't talk at all about how you did the tests. That was covered in the materials and methods section. Okay, so I really just want one or two introductory sentences and then this table. In the next video, I'm going to discuss the discussion and conclusion section.